So, hi everyone, welcome to our first annual Neuroscience Ask Me Anything. And I'm really excited about this opportunity to speak with this panel of experts. This event was hosted and sponsored by the Trend Center, or the Center for Translational Research in Neuroimaging and Data Science, which is a collaborative center across Georgia Tech, Georgia State, and Emory University. We have a number of panelists today, and I'm going to allow them to introduce themselves one by one, and then we'll start with some questions. And first, we're going to go through questions that people have, or that I've collected, and that I think would be kind of revealing the expertise of our panels and would serve general interest. And then I'm going to open it up if anyone has questions that they would like to ask in the chat. And also, I would ask just to kind of get an idea of who's here, because I invited a variety of people, but obviously I don't know everyone's names from my numerous contacts. So if you could just put like where you're from in the chat, I would really love to see where everyone's from, if you feel comfortable sharing. And also, if you could mute if you're not speaking, that would be fantastic. Um, okay, so first, I guess I will introduce Jess. Why don't you go ahead and give a little introduction of yourself?
You're going to kick it off with me. Okay, I'm Jessica Turner. I'm a professor of psychology and neuroscience at Georgia State. My research comes out of a cognitive psychology and a mathematical psychology background into cognitive neuroscience and neuroimaging. And I will study anything if there's a brain in it, preferably human. So I think I primarily focused on schizophrenia and where reality really breaks down, our internal sense of reality breaks down, but I've done a lot of different things. So hopefully that'll pique somebody's interest to ask a question.
Great. Roberta, why don't you go next?
Hi everyone, I'm Roberta. I'm a PhD student in Italy. Here I'm a visitor scholar and my research interests are about neurodevelopment and neurotypical and non-typical neurodevelopment, so in psychosis, specifically in schizophrenia and bipolar disorder.
Great. Linda, how about you?
Oh, hi, my name is Linda Larson Pryor. I am a professor at the University of Arkansas for Medical Sciences in a number of departments, psychiatry, neurology, biomedical informatics, and neuroscience. Oh, and pediatrics. And I'm the opposite of Jess. I started in imaging. I'm an electrophysiologist by training. I am an imager, so I do both EEG and fMRI imaging. My primary research interest is in sleep, but that has led me straight to cognitive neuroscience and to the sequela of chronic sleep disturbances in adults and children. And recently I've started to work in the impact of nutrition and physical activity in cognitive development in children.
Great. Ishan, why don't you go next?
Sure. Hi, everyone. I'm Ishan, currently a PhD student at Georgia Tech and a graduate research assistant at Trends Lab. My research is mainly approaching the neuroscience aspect of things from a computer science perspective. So I work on like subspace analysis in the brain to identify what subspaces in the brain are associated with a particular condition, either in health or in a non-healthy brain. So yeah, as a researcher, I'm really excited by the challenge that studying the brain poses in front of us due to its complexities.
We're so happy that you're joining us. Eloy, why don't you go next?
I'm actually an incoming PhD student at Georgia Tech, and I'm currently a visiting scholar here at the Trend Center from the Netherlands. I do research both on how we can use the brain to improve deep learning techniques and the other way around. So how we can use them to learn things from the brain, and specifically, how can we combine different neuroimaging techniques? Like, for example, structure MRI tells us something about how the brain is structured. function MRI tells us something about how the brain functioning works. How can we combine that to better determine whether someone has a psychiatric disease or how psychiatric diseases develop through their life?
Great. And Jeremy, and then Thea, you can go last.
Hi, everyone. Jeremy Bachholt. I wear a lot of hats at Trends. The main one is I'm a software engineer, kind of slash data engineer. And my role really at the center is to continue to help develop software that empowers our investigators to go after the challenges of many of these diseases. We're focusing mostly on psychiatric and neurological diseases, but also just exploring normal development. So thanks, and wonderful that you're all here.
I'm Thea Anderson. I'm a PhD student at Georgia State, and I'm studying how obesity influences Alzheimer's risk using rodent models.
Great. Well, I am really happy to have everyone here, and I really appreciate all of our experts taking the time to join us today. So I think since I made Jess introduce herself first, I won't make her answer the first question. I'd actually like to start with Linda, and a few questions that I would like to ask you, and you can just kind of pick from these in terms of what you would like to start with. First one is, why do we sleep? And the next one is, what is the most important function of sleep? And then how does not getting enough sleep affect your brain?
That's a lot of questions that are all interrelated. So we'll start with the central question, which is why do we sleep? And my favorite answer, we don't know. We only know that it's critical. So we do know that if you don't sleep, especially if you don't sleep and you're a mouse, you die. If you don't sleep and you are a human and you truly don't sleep at all, as happens with fatal familial insomnia, you will also die. But it takes a lot longer in humans, as you might expect. So on the way towards that awful end point, and before you get there, you will fall asleep, you will have a number of sequelae that are entirely reversible as soon as you go to sleep. So you'll start to get foggy in your head, you'll stop being able to attend things very well, you'll begin to lose memory function even if you're 20, sorry to tell you that, guys. So those kinds of things will happen. If you're older, it could be even more substantial as a problem. The other interesting thing about sleep is that we know that sleep disturbances are associated with every neurodegenerative and neuropsychological disease, all of them. So perhaps you are most familiar with Alzheimer's disease with sundowners, with the fact that Alzheimer's disease patients have disrupted sleep, with our understanding that the lymphatic system works best when we are asleep and therefore clears neurotoxins best when we sleep. And that's one explanation, one thought on actually the development of Alzheimer's disease, that we're just literally, sleep is critical for clearing toxins from the brain. And if they don't get cleared, then they land ultimately with chronic disruptions. If you would mute, that would help. Whoever's talking right now. So it can lead to disastrous sequelae. So there's a short answer to your question. I could go on for hours.
Well, hopefully we'll have the opportunity to do so when we're done. That was a really great explanation. And it's hard as a scientist to be able to kind of convey such complex studies in a short amount of time. And you did a great job.
Yeah, sorry, just to add to it, I think I recently read a book called Why We Sleep by Matthew Walker, which is a kind of a good book to give an overarching view of how sleep functions and how it gets affected by so many things.
And there's another book by William Dement, who's perhaps one of the fathers of modern sleep neurobiology, and it is called The Promise of Sleep. Not too expensive on Amazon, really fun read.
Great. And actually, if you could put that in the chat, I would like to share some of the things that we talk about with people, just so that people kind of have a list to go through in terms of, things to look at and resources to access after we're done today.
There was a wonderful book on the family that couldn't sleep about inherited familial.
There are 800 familiar images.
Yeah, I was hoping Linda could either give me a thumbs up or a thumbs down on whether I can trust it, because I read it and I thought it was absolutely phenomenal. It was highly informative about prions and things.
I'm afraid I haven't read it, but it is a familiar.
For that visits I'm doing tomorrow.
Someone is talking constantly. With maybe you don't know that you're unmuted.
I changed the settings now, so hopefully anyone who's new, but please remind all of our participants to mute.
It's like you're almost. I told them, I was like, look, I'm almost done. I only have health left. Bio. I just told them I don't need help with bio anymore. And like, I'm probably
anyway.
now.
Anyway, it is a familial disorder.
If it's only 1 or 2 classes, then I'm not going to make you pay and charge you. So
excuse me. Sorry, everyone. Whoever is speaking, we can hear your conversation.
Then it's a separate story. Then you'll have.
And I don't know. I'm trying to look at who is unmuted. And I now changed the settings.
Like this is like my last health unit. Physics, this is my last unit. Bio, we have 1 more unit, but like I already did A's on the tests.
There's someone talking about their bio unit and their health unit. Can you please mute yourself?
And I'm a senior now. I'm like, whatever.
I don't know. So it looks like everyone's muted, so I don't know what to do. Okay, let me just try to look one more time and then we'll keep going, I guess.
No, first year I'm taking the easiest classes possible. I know, I'm going to take the easiest classes possible.
Wow, it's
just
interesting.
To get into it.
It's as though
I already know that.
Wow.
Anna? Yeah. Tomorrow, Eugene, coming.
I'm wondering if this is like a technical thing because everyone is muted.
I send out too. Honors program.
So I'm wondering if we should like hop off and come back on because it looks like everyone is muted.
Yeah, I think this is getting a crosstalk from some other device.
Or
I think this is someone's
phone.
phone.
Themselves on hold or something. Very strange.
Is it still going?
Yeah, it is.
Yeah, it really looks like we're in the middle of somebody's conversation.
Okay, so I'm wondering if we should just we're gonna get off here, I guess, and then come right back onto the same link. Should be, I think there's a way, or why don't I post the link in the chat and then we can all come right back here.
Okay.
Yeah.
Let me do this. I'll do this real quick.
Strange.
So, Miji, you don't think that we can use the same link that you had originally?
I don't think the chat saves.
No, it doesn't save. That's the trick.
I'm gonna save it for. I'm gonna copy it and save it for everyone. Okay.
And of course, now it's quiet.
Okay.
So we should just hop off and then hop back on. Same link.
Yeah, I'm actually just gonna stay here because it doesn't sound like it's happening anymore. So
Yeah.
I'm just gonna stay here and wait for people to come back on, and then hopefully we can continue because that was really weird. Because I muted every single person.
Yeah.
I've never had this happen before.
I don't know what would cause that.
Yeah. Okay, I'll get everyone else. You guys can just stay, I think. I think now it's over.
It's not happening now, so
Right.
whoever it is either fixed it or it's not happening.
I've never heard of it. That was so weird.
Yeah. Okay, and I have it as mute on entry so that when you come in automatically, you have to actually unmute yourself. So hopefully this works now.
So, fun fact, if you hold the space button down, you unmute yourself just long enough to get muted again when you're done talking.
That's great, and I actually try to use that because like I feel like I'm using a walkie-talkie when I do that. Okay. So it looks like we can pick up where we left off. So you guys, I actually have saved the I've saved the chat so far. So if this happens again, I can just copy it and bring it back in. But I think we should move on to our next question, which is so this is actually a question that maybe Roberta and Jess can kind of answer, maybe each give their opinions. So, because I know Jess is really familiar with psychosis as well, and that's what Roberta is studying. So the question that I had was, how is a person's brain with psychosis different than someone who doesn't have psychosis? I see Jess laughing.
Well, that's
because
right up
it's
there
a
with
complicated
REM
question.
sleep. I'll focus on schizophrenia because I am not a clinician and I'm not as aware of non-schizophrenic psychosis other than bipolar disorder. But I'm actually in the middle of reading a book called The Mad Doctors. I think it's basically about the history of the rise of psychiatry start as a medical profession starting in the late 1700s. And it was actually a really interesting, the very first chapter is all about this one fellow who was basically making the argument that it is it was a medical problem, that mental health and madness at the time in England was considered in many cases a moral problem rather than a mental problem. And he claimed, no, it's actually something that should be under the purview of medical doctors. But that said, of course, we always have to, whenever I'm presenting to, undergrads or the students that I tend to talk to, I always have to point out that there isn't an X-ray that I can give you. There isn't a blood draw that I can give you that says you have schizophrenia. It has to be at the moment identified through symptoms and everything else. But once we actually get enough individuals with that diagnostic category together, we do actually see differences in their brain, which is exactly the kind of thing that Eloy is working on, or was it Ishan who was working on, trying to predict, from structures in the brain or patterns in the brain, whether or not someone is considered to have psychosis or not. And there's certainly been a lot of work from huge groups like the Naples group and others to say, can we predict who's going to develop it before, rather than waiting until they have the diagnosis and then figuring out what's different in their brain? Can we look at their brain ahead of time and try to figure it out, which has not proven to be particularly useful, unfortunately. Because our current ability to measure the brain, I think, is our limiting factor. I think there will come a point where we can measure what's going on in the brain and say, yeah, this person's, serotonin or dopamine or whatever is not functioning the way it should, and it's putting them at risk. But at the moment, it's almost invisible until you look down the road and say, okay, somebody with schizophrenia, you've got larger ventricles, you've got smaller hippocampus, and you can see some things along the way. And obviously there are huge differences when you look at enough individuals with schizophrenia. But it's not like we can say, oh yeah, there's a particular break between this point and this point that makes you schizophrenic. So hopefully everybody's working on fixing that.
Yeah,
And
also because we can talk about a spectrum of psychosis and not a disorder itself. So there are many symptoms. Some people could have some symptoms, other not. Also measure by brain measures these futures is of course difficult, but it's challenging, is what makes all this field of research interesting, I think.
And I think this would be a great point for Ishan and Eloy to kind of talk about, they use, from what they have said, they've used a lot of different modal, which to someone who doesn't do neuroimaging probably doesn't make a lot of sense, but basically a lot of different ways to look at the brain to determine what is going to predict someone who, maybe doesn't have psychosis right now to developing psychosis or things like that. So do Eloy and Ishan, you each want to share kind of how your research fits into what we've been talking about?
Yeah, I don't mind starting. So I think especially because it's such a complex disease, and also because oftentimes people with, for example, schizophrenia, they are being the way someone determines that they have schizophrenia is generally not through a brain scan, right? Although we do know that probably the differences we see in behavior in people with schizophrenia, or the symptoms we see clinically with schizophrenia, are probably in some way reflected in their brain functioning or the way their brain looks like on a neuroimaging scan. But it's very complex for doctors to just look at a scan and say, oh yeah, this obviously is someone with schizophrenia or not. These types of things are only possible with things like dementia, where you can actually, people that have like dementia far into their dementia, you can see that their brain has shrunk, like in the gray matter of the borders of their brain is sort of have some shrinkage. But with schizophrenia, it's super complex, right? Like Jessica talked about, it's not like one thing. It's like you have schizophrenia. It's a very complex disease, and this holds also for diseases like autism spectrum disorder, which are also on a spectrum, and it's kind of hard to say, and so it's hard for someone to look at an image and say, okay, this person has, this disorder. So what we try to do is teach machines to look for patterns in the data that we have to find maybe patterns that we overlooked as if that humans may overlook if they look at scans.
Yes, I think Eloy sums it up pretty well. I'll just add a couple of quick points. I think in terms of certain disorders like Alzheimer's disease, it's a very structural disorder. Like it affects the structure of the brain. So when I say it affects the structure of the brain, it also means that whatever machine learning or computer science techniques you would use to find patterns in the brain to detect it, it would be easier to just look at the structural data in the brain and be able to some extent predict whether a person with just mild cognitive impairment would also develop Alzheimer's disease going forward. But I think with a lot of psychosis disorders, it's kind of hard because they're very functional in nature. A lot of studies have pointed that, predicting or like diagnosing them through function, looking at functional data and functional patterns in the brain is easier compared to just looking at the structure. So I think in that sense, it's even the diagnosis, developing a foolproof diagnosis is still a work in progress. And I think apart from Alzheimer's, I think in case of psychosis, it's as of now, it's not very clear on how to predict it beforehand, like before it happens. There are some studies which do work on like developmental data sets, or like adolescent data sets to try to predict beforehand whether a given person is susceptible to a certain disorder in the spectrum. But it's, at least in my limited knowledge, it seems like it's a work in progress still. I think the reason for that is, like, imagine if you were to think of like so many, in the popular media, we come across so many studies that regarding the body, right? Like this kind of a lifestyle or this kind of a body feature is associated with a given chronic disease. So it's hard to do it even at the level of the larger body, where we still have a way to isolate the organ and study it really well. I think in the brain, it's with MRI and fMRI, which are the techniques, it's not very easy to study the quick functional changes in the brain at this stage. Like, yeah, someone could add to my points. But
Well, I
that's all I
actually
have to
think
say.
that this would be a great place for Thea to come in and talk about her research using animal models, and how, for those of us that, aren't career scientists, which I know many of us are, what are some of the advantages and disadvantages of using animals to study things in this way versus using neuroimaging techniques?
So I was about to actually chime in and say, hey, that's one of the reasons why we use animal models, because you get inside that black box, and you can isolate individual molecular interactions, and you can look at what's happening downstream as a result of that. So usually you would start with like a gene scan to look at what is different in people with a certain disease. You look for common pathways, and then you put those into an animal, that you can then look at the consequences of this, look at interventions that prevent these consequences. And I think it's really awesome being able to actually take the brains out of my subjects and really investigate what's happening with them.
Yeah, I think it's kind of I think many people who aren't scientists don't understand the advantages of studying animal models and the kind of convenience that it comes with. But I think that it definitely allows you to control for a lot of things and really isolate what it is that you're particularly interested in manipulating. So,
Yeah, are you doing the multi-electrode recording or just other measures?
No, I'm doing, well, with my current project, I'm doing behavioral data, and I'm going to be looking at amyloid oligomerization. Um, it's 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 the beginning. It's the beginning. So my goal is actually to put sensors in the brain of an awake behaving animal and look at like so there's like a little tunnel, it's a little cannula, the sensor goes in and changes color, and records an electrical signal based on the amount of oxygen and blood sugar and neurotransmitter that it detects. And that's my ultimate goal. So ask me about that maybe in 2 years. Hopefully I'll have something nice to share about that then.
We'll be back.
Great. So I kind of want to shift. So we talked about, how we're using neuroimaging data to kind of face these really difficult problems, in particular in psychosis. And I would actually like Jeremy, kind of our resident data science expert, going from the small animal models to, large big data, which I know that a lot of people are more kind of familiar with what large data sets are and what is big data in this current era of science. And I was just wondering, Jeremy, if you could talk a little bit about some of the challenges and also some of the advantages of using really, really large data sets in research.
Well, first off, thanks for the question. And yeah, I mean, I can't speak to the exact correct statistics, but when we want to do things like take neuroimaging and line it up with genetic results and fuse those two and analyze them together, we need very large data sets. And we now have access to very large data sets, but it doesn't mean we have the tools and the knowledge all in place to actually handle that scale. So one of the things that we've done right away at Trends is we've tried to address the scale issue by moving all of our data effectively to the cloud. And all we mean by that is you can think of the cloud as somebody else's computers, right? We don't actually have to have all of those computers on site. We basically rent them from Amazon. And so it takes a lot of thought and effort to move all of this data. And it really is a moving the data. And so we've got to have consistent formats. We've got to understand where the data comes from. We've got to understand how it varies. And it all kind of boils down to standardizing this data, having a shared vocabulary, and having a way of explaining and sharing the data with others. And finally, a big issue for us to deal with now is privacy. Just, genetic data is can reveal sort of who it is. And so we've got to be careful with data and trust and secure the data. And so we're working on all of those issues here at Trends, and we're hopefully helping each other, training each other on ways to follow best practices. So at least as a first pass at that question, that's what I've got.
Thanks. That was excellent and very informative. Okay, so now we are right in the halfway mark, and I have some more questions that I would like to ask everyone. These are kind of more general questions that can be focused on careers and also kind of the state of science and your more big picture questions. But I would like to give everyone the opportunity, if they would like to pose some questions in the chat. I would love for our experts to be able to answer some of those. So I'll go ahead and ask some of these questions, but I will also, if interesting questions come in, I will make sure that those get answered. Okay.
Maria, may I make a comment very briefly?
Please.
Thanks. So regarding psychosis, there's a tool that seems to be overlooked, magnetic resonance spectroscopy, excuse me, that assesses some of the neurometabolites in the brain. And a lot of work's been done in looking at psychosis with MRS and teasing out a lot of neurochemical changes, particularly with glutamate and glutamine. And most recently, there are a lot of research reports of correlations with glutamine and psychotic symptoms as assessed by various neuropsych tests. But one of the big problems with studying psychosis with any tool is that the symptoms and the subject very quickly. You have first break, you have chronic, you have people who are medication naive, you have people who have been on medication, and we know that medication changes the neurochemistry of the brain. So it's a moving target. And that's where the advantage of big data and normalization comes in, as Jeremy stated, in having enough subjects in your population with similar symptoms and measurements that you can actually attribute to a particular state of psychosis, or to the beginning of psychosis, or to chronic psychosis that doesn't respond to any medication. Anyway, I just thought I'd throw that out there.
Thanks. And would you just give everyone, since you're not official panelists, but we're really happy to have your comments, just a little, just your name so everyone knows who you are.
Yeah, I'm sorry. I'm not an official panelist. I just joined in to follow along. My name is Helen Petropoulos. I've been working with magnetic resonance research for quite a long time. I work for the Trends department. And thanks for giving me a few minutes of your time.
Thank you. We're always glad to have our participation from everyone, so that's great.
Can I follow up a question?
Oh, sorry. Go ahead, Jess.
Okay. Oh, no, I just had a question for Helen, because of course the spectroscopy that I used to do, you were looking at a massive chunk of the brain. So you weren't getting very spatially resolved sort of information about these metabolites and the chemistry. What's the best we're doing these days with sort of the latest 3Ts? Obviously, if you get up to 7T, you can do better. But what's the spatial resolution on the current spectroscopy?
Oh, that's a great question. So 3T gives you much better resolution in the area of the peaks that the glutamate, glutamine, and GABA peaks, which have overlapping resonances. So the higher the field, the better the resolution is, and the better you can fit that spectroscopy. With 1.5 Tesla, I apologize for the dog. With 1.5 Tesla, it was very difficult to get a glutamate and glutamine separation. Now at 3 Tesla, I think people can accept a measurement of glutamine in and of itself. But for something like GABA, for example, you have to have special spectral editing techniques in order to capture something like GABA, which is equally important in the study of schizophrenia and the neurochemistry. But at 3T, very definitely, there's a lot of data on glutamatergic and glutaminergic changes in the brain at various stages of psychosis. I think what's interesting is that they see different changes with first break and medication naive subjects than they do with chronic subjects. So it's definitely much better now, much more informative now. And I think as 7T becomes a standard, that's really going to go into something that's very useful as a tool. I don't know if that answered your question.
No, that's actually very helpful. I was thinking more about the area of the brain that you have to image in order to get a decent signal.
I see. So anterior cingulate is very important.
Everybody loves the anterior cingulate.
Everybody loves the
Anterior
anterior
cingulate does everything.
It does everything. And it's also a great place to place a spectroscopic voxel because some of the other, brainstem areas, you run into problems with tissue and air interfaces that are not helpful in looking at your spectroscopic results. Most of the work that I've done involves the anterior cingulate. Certainly there are, many places in the schizophrenic brain that are going to give you some interesting analysis results. But anterior cingulate seems to be consistently a very good place to place your voxel.
And what is the size of your voxel? Are we talking centimeter scale, millimeter scale?
That's another very good question. It's a centimeter scale. You can do a spectroscopic imaging, make an image, but your voxels have to be much bigger in order to get enough resolution to get a decent signal for things like NAA and other neurometabolites, because they're clearly not as abundant as water is. But typically for single voxel, I think 8 by 8 by 8 millimeters is a decent size.
So if I could chime in on this, I just think it's interesting that you asked that question, Dr. Turner, because I've seen a lot of studies that are getting more and more popular using the chemical shift paradigm for spectroscopy, which is where they're just doing like whole brain MRS. And while I think that may be interesting, I have mixed feelings about the pros and cons versus something targeted versus something whole brain.
Well, there are differences between doing the whole brain and doing something very localized, particularly your resolution. You're not going to get as good a data set from an image as you are from placing a larger voxel in a localized area. But whole brain spectroscopy gives you a bird's eye view of major changes and, perhaps an idea of which areas you should be targeting for higher resolution. And spectroscopic imaging across the entire brain is also getting much better as far as the technology. It's much better than it was just a few years ago. So in time, hopefully you can acquire spectroscopic voxels from the entire brain and get a reliable estimate of some of these neurochemicals. But yeah, there are going to be differences than looking at a localized region.
Well, great. This has been a nice little MRS tangent, which is great. I feel like this kind of discussion is what these are kinds of things that are supposed to kind of open up when we talk about these things.
So
Linda,
Maria, could I hop in on another?
yes,
kind
I
of
was
a-
going to say, yes, you were going to say something, so please.
So Helen has brought up something important, which is we all tend to get a little bit idiosyncratic about the methods that we use to study the brain. And physics has taught us that that's usually a bad thing to do, is to fall in love with your technique and not be aware of multiple other techniques available to you. So we have electrophysiological techniques that are non-invasive. EEG and MEG are two of them. And they also provide very useful data. And we've completely ignored functional imaging, which goes beyond fMRI and includes things like PET and TMS and multiple other methodologies. And all of those have been at one time or another, with greater or lesser utility at the time they were first administered, been used to study brain-related disorders. Of course, those include neurodegenerative disorders, developmental disorders, TBI, for example, currently COVID. All kinds of things can cause disruptions of the brain. And I think one of the things to keep in mind with a lot of what we do is that there are similarities. When the brain breaks, it tends to break in similar ways. And so we have the classic P300 conundrum, which some of you are probably familiar with, which is everybody studied the P300 and what did they discover? It's abnormal in every disease we've got. So it's completely non-specific, but it is telling you something about the brain, something that's important, which is that the brain tends to break in fairly straightforward ways. And it may break those ways for different reasons, but it tends to do that. And I think sometimes we ignore that. I think sometimes we just lose sight of the fact that that happens, and we think, oh, look, we found this one thing that happens, and that must be what happens in this disease. And we can fall into the same trap that the P300 did for, like, what, just a decade?
or hippocampal atrophy. You know, every disease has hippocampal problems, no matter what you do.
No,
but everybody thinks it's somehow specific to their disorder.
But that's a really good point. And that's why you have to use multimodal approaches and look at, data from a lot of different perspectives and find an effective way to tie everything together in a way that makes sense. And I know that that's a huge challenge, but I think that's also one of the goals of the Trends team. I'm not sure specifically who's working on it, but very good point that you have to look at the whole kit and caboodle to make an intelligent decision about what's really happening in a very complicated disease.
And actually, we did have a question that was a really great discussion and some great kind of scientific big picture takeaways. I actually did get a question, and this is for Thea. And what is the biggest challenge? This is more of a general question. What is the biggest challenge with having animals as subjects?
I guess it would have to be the complexity of their behavioral repertoire. We have to use sort of crude proxies. Like if it's mice, then we'll say, oh, they have hyperlocomotion. Maybe they're showing signs that might be somewhat related to psychosis. Like the classic thing that people always say is you can't ask a mouse if it's hallucinating. Which you can't, but you can also look at molecular interactions in ways that you can't with people. And also you can look at specific time points. Post-mortem brains, you just kind of get what you get. Whereas, not to be gruesome, but with animals, you can get it whenever you want it. The challenge right now, I'm using rats, and previously I worked with mice, and the rats are surprisingly a lot smarter. I think, yeah, because they're just like little rodents kind of scurrying around. You have to use a whole lot more of them to get like a consistent behavioral phenotype. But yeah, I mean, I love working with animals. I don't know how you guys managed to. I mean, I understand, like, you can see so many things with images. You can ask the people what they're doing, how they're feeling, what their affect is. But I just feel so much more powerful being able to completely manipulate their environment and the life that they experience.
Well, that's actually a really great point, and I'm wondering if maybe Eloy and Ishan, and I think to some extent Jeremy too, have had some experience with kind of new data analysis approaches, such as deep learning, that can be, really informative in a way that animal models are not. And so I'm wondering if you would like to speak, if one of you three would like to speak a little bit about how, kind of in contrast to what Thea was saying about manipulating things, how have you developed approaches to get the most out of neuroimaging data in situations where you have no control over what's going on?
I can add a real quick point to this. I think deep learning, like it has revolutionized so many fields, is also like being used very widely in the field of neuroimaging data analysis. And I think there was a recent study from the Trends Lab that also compares deep learning thoroughly in terms of diagnosis with the traditional machine learning methods. So it's like deep learning very clearly outperforms when it comes to like scan-based diagnostics. I mean, one of the potential issues with deep learning is that it's very hard to track like how it is working exactly. It's theoretically not easy to, pinpoint what worked out exactly so that we can trace back the insights of the whole patterns. But in terms of performing, it's doing much better. Yeah. So
It's actually
one part of my research. Sorry, were you saying something?
I was quickly going to add that the way we also look at deep learning models is kind of the same way that Thea looks at animal models in some way. We try to perturb like neurons in a deep learning model to see what that neuron in the deep learning model has learned. So we kind of come back to like, we use these models, and then to know what they learn, we use kind of the animal model techniques on them, because we can, like Tia said, manipulate their whole, like both the neurons inside of it, but also the whole sort of environment around that deep learning model. So I thought that was a funny.
Yeah, that's kind of a cool point, because the deep learning models are, after all, like they're more like artificial brains, you can say. Not as big as even a rat, though, but still, yeah. I think manipulating them, and I think one of my thoughts is that it is very possible going forward to to manipulate deep learning models and understand them better, to provide more insights into the inner workings of them. One of my recent work also explored like multimodal deep learning models, and then trying to pinpoint what features or what subspaces in the brain are more complex or have more information about a given diagnosis. Like in my case, it was schizophrenia. So deep learning is, I think, there's a lot to be done going forward, especially in terms of integrating the deep learning models and tweaking them.
Yeah. What I also want to add to that, and Jeremy touched upon this, we have a lot of data, and we just don't know what to do with all of that data, as in it's too much to use all at once. And that is where these sort of computers models come in, where we can say, we can't overlook all of this, like it may be like terabytes of data. And if a human would look at that and try to piece all of the pieces together, it may take years to get, obviously, to piece all of the pieces together. That would take a very long time. And we try to use these models, these computer models, to do that for us, and then sort of afterwards look back and say, okay, well, what pieces did it piece together, and does that mean anything to us? And so for me, it's mostly about, we have like so much data that we've gathered throughout the years with neuroimaging techniques, but it's too much for one person to look at all at the same time And that's where I feel deep learning also comes in, is to make use of all of the data we have, sort of reduce it down to a biomarker, for example, that we can use to test on for certain diseases, but also to look at what if you could piece all the data together, what pieces do you select, kind of.
Yeah, in some ways it feels like science has kind of just done this full circle, and that, like, you know, we used to think that there were these unexplained factors and like magic that was like controlling things and doing things, and now we put things into the computer and we're like, well, the computer does magic, and then we get this cool thing.
So
The only thing I would add, oops, sorry, go ahead.
Um, I'm going to put forward something that I've been thinking about a lot. So the disease I work in is a neurodegenerative disorder, and it is a neurodegenerative disorder that affects far more than the system for which it was originally defined. So it was originally defined as a motor system disorder. It is not just a motor system disorder, it's also a cognitive disorder, it's an autonomic disorder, it's a sleep disorder. It has all of those factors. And so one of the things, and I think someone touched on this earlier, that I think, speaking of bringing things full circle, is we've torn everything down, taken it to the reductionist heaven of genetics and microbiomics and metabolomics and proteomics, et cetera, et cetera, et cetera. And we're in danger of forgetting that it's an interlocking system. So in fact, the brain does not live in isolation any more than the kidney and liver live in isolation. And they're all interacting one with the other. If you affect one, you affect them all. And I think sometimes, especially with really complicated disorders, which is what we want to study, is these really complicated disorders that affect human beings and their quality of life, we do so to our detriment. At least in the back of our heads, we have to keep in mind that they're really not completely isolated from one another.
Jeremy, did you have something you wanted to add? Because that's a really great point.
Yeah, no, that was a great point. Thank you for that. And all I was going to add is just one of the, you know, what can we do going forward was part of my earlier question. And one of the nice things to witness, watch, help out with is that many of these machine learning models are now becoming or have a goal of being explainable. That's the new term for it. And it's going to be very cool to utilize these models where you can hopefully go back and try to explain why a particular thing is happening, right? You know, a lot of them have been just black boxes. This is the result, and you just sort of have to use it. But being able to explain why, I think, is going to be incredibly helpful.
If I could add something to what Linda was saying, because I was sort of seeing the same pattern, because Linda's making a fantastic point that the brain doesn't, just like no part of the brain works in isolation, the brain doesn't work in isolation, and the humans don't work in isolation, and you end up in sociology before you've gone too far. But I was also thinking from the point of view of the animal models versus the big data kind of approach, and the data mining and machine learning kinds of approaches, is we've got on the one hand an extremely controlled environment where you can tweak things and manipulate them and know exactly what's going on. And then we have the hypothesis driven, let's look at a region and see what's going on. And then we've got the sort of whole brain. I'm not going to go in with any a priori hypotheses. You tell me what the best fit is for quote unquote what's going on. And we all have to work together. You know, we can't just say my way is the right way to do things, or the questions that I can ask are somehow the most important. We do have to pull from each other's findings back and forth.
Yes, and I think that this has been, you know, I think that any scientist will, or I would say most scientists that are really aware of kind of the big picture will be able to kind of step back and say, you know, this is what I think and this is what's going on, but we have to step back and remember all these things, which are all the other points that everyone has brought up today. So this has been a really great, fruitful discussion. So I wanted to kind of finish with one last question for each person. Obviously, if any of our panelists have to leave at 5, I totally understand. But I would like for everyone to just give a really quick answer of one of these 3 questions, not all of them. Just pick one that you like the best, which is, what can I do to take care of my brain? And what are some of the big questions in neuroscience and neuroimaging as a whole that are the most interesting to you? And what do you think would be the most interesting development in the field in the next 10 years? Or what would you like to happen? You know, okay, in the next 10 years, I would like to have this question answered. So one of those 3, everyone can answer. And I'm going to start. I actually will start with, I'm just going to pick a random person. Let's start with Linda, and I'll call people.
I get to answer 2 because I can answer one so fast. One of the best things you can do for your brain is get enough sleep. Then on to what do I think is like something really interesting and a huge problem in neuroimaging that we're all dealing with, and that is our ability to share data across sites. So we can't have big data. Jeremy's already commented on this, but I think it's well worth reiterating. We can't have big data. We can't share our information if we do not harmonize that data. So we all necessarily use the same scanners. Unfortunately, we do not all necessarily use the same scan protocols or the same specific behavioral protocols for anything that we do, or the same task protocols. We don't even really know, and Jessica's very familiar with this. I was hoping she would get into ontologies at some point, but she says no. One of the things that I think we have to recognize is that when we talk about a specific cognitive domain, we may all use different ways to test that domain, and we may not be testing the same thing. So the assumption that just because I'm looking at executive function, for example, and I happen to be looking at dorsolateral prefrontal cortex somehow, everybody else who looks at dorsolateral prefrontal cortex and executive function is looking at exactly what I'm looking at is just not going to work. So I think that's a huge challenge particularly for the neuroimaging field, but I think I would say for neuroscience a little bit more generally, because we can go to Thea's work. She's going to be working at the same thing, but she's going to be looking at a different organism whose brain is not exactly the same. We're making assumptions about the interrelationships between those parts of the brain, but they may not be completely true. So I just think that's one of the huge problems that we're going to have to deal with going forward.
That was a fantastic answer. Okay, actually, since she mentioned you, she called you out, Thea, so now you have to go next.
Hi, I agree that that finding ways to bridge the animal and human is really, 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 really important, because if we can't verify that what we're doing has relevance, then we're just doing sadistic things for no point at all, and that would be horrible. I want to answer the first question, what's the best thing you can do for your brain? It's the really boring things that you already know. Get enough sleep, exercise, and don't drink alcohol. Exercise in particular, though, I've been reading these things about how it activates cellular pathways that make your brain better at taking energy from the glucose in your blood and metabolizing it. And it activates pathways that, well, okay, in rodents that have access to run on a treadmill, the ones with the exercise, their brains more resemble younger. And they also get the same thing with intermittent fasting, but I mean, that's kind of mixed results when you come to humans. Although I'm sure everyone has very strong opinions about it, if you have an opinion about it at all. But yeah, and alcohol, it does very bad things to your brain. It shrinks your brain volume, it drains the water out of it. You're mostly water. So all the things that you already know are really the things that you should do to keep your brain healthy.
You're not going to recommend blood transfusions from young people?
Yeah,
we were all very hopeful about that whole vampire movement a few years ago.
if you are powerful and you live in a regime that doesn't value human life, then you can get blood transfusions from young people and it will keep you quasi-immortal as long as you can keep getting that young blood. That's something that hopefully will never translate to humans.
Hopefully not the most interesting technological development in the next 10 years.
I can go next.
Great.
So I would like to answer two questions because one of them is real quick. Like one big question in neuroscience. Personally, that one that excites me is consciousness. I think like I studied a little bit of philosophy in my undergrad, and so I've always been very curious, like what are the neuroscientific correlates or neuronal correlates of consciousness where it happens in the brain, which I think is like, it's indeed a very big question in neuroscience. But secondly, I think about the first question, keeping the brain healthy, I think there are so many new techniques or new studies that are pointing to various things associated with brain health. Like I think Thea mentioned alcohol, right? And I think there are some lifestyle changes which are like in terms of sleep, and some in terms of like even when you're awake in the day, taking care of certain things, just like how we take care of our body. More recently, like personally, I like to think of it in terms of how the world recently is going through a cycle of lifestyle diseases related to the body. Like for example, obesity. It has affected so many lives in the world. Similarly, like I think in the mental domain, there are certain lifestyle changes which are affecting the brain health a lot recently, especially in the digital world, we could say. So I feel like amongst all this, I personally like to reflect like what could be a good diet for my brain in terms of the kind of interactions I have or the kind of activities I do throughout the day. Am I like consuming healthy things when it comes to mental consumption in terms of thoughts and emotions? So that's one. And one more thing, last thing I would like to add is like I think meditation, various kinds of meditation techniques are available, yoga techniques. They also help, at least personally, they help me calm my brain. And I think there also have been studies which establish that they're very helpful in various aspects of mental health.
Great. And some of those, from my understanding, some of those studies that have looked at meditation and health are actually using some of the like trends developed data storage and data analysis methods. So I think that's pretty cool. So we can say we have some of the data to support that. We literally do. Okay, so now I guess I will turn it over to Roberta, and she can answer, and then we'll do Jess after that. And then we'll do, you don't have to remember this order, more for me, and then Eloy and Jeremy can go next. So Roberta, take it away.
So answering the first question, since I'm Italian, I can suggest a Mediterranean diet because also what we eat is important for our brain, for all the nutraceutical perspectives, or the substance that are present in our food is important for our brain health. And then answering the second question, my personal interest, and I think is something of general interest, could be he's trying to find a way through complex models and using big data that allow us to track and predict which factors could be important to determine a psychosis or a particular psychological condition So in order to prevent it and through some treatment, not necessarily using drugs, but also behavioral treatment or some strategies that could help and yeah, could help people to live better and prevent psychological suffering.
That's great. And it looks like your work is trying to contribute to that. So that's you're actualizing your own desires. So that's great. Okay, let's have Eloy answer.
So my personal goal actually is more of a societal one. I think that by using deep learning and as computers and those kind of things become cheaper, I hope that it contributes also to more time for doctors to spend with their patients, more fair healthcare to more people, make it more available to different people, make healthcare cheaper, make sure that it's not this thing that is only available in rich countries, the newest techniques. It's not only available to people that can pay for the treatment they want. Make it something that is like a human right all across sort of the world. And I hope that, with the growth of technological advancement, I don't think technological advancement should take over the role of any doctor in any way, but I hope that by making it easier for doctors to work together with things like machine learning models or computer models, we can make healthcare more accessible to everybody.
That's so great. And I think that, it seems like everyone is kind of the things that they're interested in and the questions that they would like to have answered, they're also working towards those questions, which I think is fantastic. And then I guess we'll have Jeremy go. And I think it's funny because, of course, I tell a bunch of scientists, like, please answer one question. Everybody answers two. But that's typical, and I think it's great. So
I'm going to be guilty of answering two. I just participated in a methylation study where we essentially were able to pick up a 2-week change in smoking just from the methylation results. And so if that doesn't encourage we not to smoke, I don't know what does. Anyway, substance use or abuse is not good for us and certainly not good for our brains. I want to just empower open science. I want us to share data. I want us to share our code. I want us to share our time and thoughts and encourage ourselves as scientists and even the community in just exploring all of these neurological diseases and learning whatever knowledge we can to aid and improve and design new treatments. So I will be all over it.
great. That is fantastic. Well, Jess, you were supposed to go, I think. Did we skip you?
Oh, that's fine. Don't
Yeah,
worry about
yeah,
me.
well, of course.
Well, I'm going to answer neither question. I'm going to use the scientist's prerogative to say, so, some of the things that are on the top of my head in terms of some of the most useful developments. One is that in many cases, we always wish we had a baseline, right? We always wish somebody shows up with a disorder and you say, well, if I knew what you looked like beforehand, I'd be much better off. So I think initiatives like the ABCD paradigm, I would ideally like to see everybody scanned every year, as part of your physical checkup. Probably completely irrelevant, but you never know when it's going to be relevant. And the ABCD trying to collect, however many tens of thousands of kids and follow them longitudinally will be invaluable for exactly that kind of developments. And in 10 years, we'll be in a really good spot. Just like it took 10 years to get a rough draft of the genome, and we're still working on it, and things are still moving forward. It takes an incredible investment of time to really have a breakthrough on these things. So that was one thing that's going on. The second thing is the large-scale international collaborations for imaging and genetics, particularly genetics. I just gave a presentation Tuesday about a paper that came out, I guess, last year where they're finally talking about GWAS across non-Caucasian, non-Chinese populations, and actually attacking diversity just head on, saying, okay, if you take people whose genomes we've always avoided for the last 20 years because we didn't know what to do with them, we now are at a point where we have enough people and we have enough technology and we have enough mathematical sophistication, et cetera, that we can start to put these things together and say, yeah, there are some areas of the genome that have common risk. If you're human, this area of the genome is where some of those loci are that give you risk for schizophrenia. But depending on your particular genetic background, there may be other SNPs or other regions that are important for risk. And that to me is unbelievably optimistic, to be able to say we really do work better together as a worldwide effort to address these worldwide problems. It's just incredibly promising, I think.
Great. Well, I totally agree with pretty much everything the panelists have said. I think that I really appreciate you guys taking your time to talk to me today and talk to everyone. And I will definitely put this recording up. The Trend Center will be making a YouTube channel, and this will be the first thing that goes up there. And so hoping to do more of these in the future. And if anyone is interested in participating, I'm thinking of doing like kind of big data questions, psychosis ones, because we had a lot of really interesting discussions that I wish we had kind of more time to flesh out and look for particularly targeted audiences. Like, okay, we're going to talk to these elementary school kids. We're going to talk to a bunch of, 50 to 60-year-old people and just try to get some different perspectives on things. And if everyone's interested, I would love to have every one of you back. So thanks so much.
Thanks
Thank
for
you.
organizing this.
Yeah, thanks for organizing
Super
it.
fun. Thanks
Yeah,
for
it was
putting
really
this
fun.
together.
Thanks, everyone.
Thanks, everyone.